And we have our final speaker of the morning, Kevin Healy from the University of California at Berkeley. He is a professor of bioengineering and material science in the engineering faculty at Berkeley. And his laboratory explores the interface between biology and material sciences, particularly aims to develop engineered systems to explore fundamental biologic phenomena and new applications in translational medicine. And he's going to present on his own model of the human heart on a chip for disease modeling and developing new strategies to treat cardiac disease. Kevin, welcome.
Thank you.
To your talk.
It's wonderful to be back in Toronto, seeing old friends and colleagues. Thank you for the generous invitation to come speak. It's a wonderful meeting so far. Took a lot of notes, learned a lot from some of the clinical presentations. Thank you, Chairman, for the introduction. I think I can buy in a little bit on Melissa's introduction, so I'll go a little fast. I'll give you the outline of my talk, which will be a short introduction built upon by what you reviewed or have seen the last few days. I'll talk about our own chip, which goes in a direction that is to minimize the cardiac organoid system onto essentially a computer-type platform. And the last part will probably be the last 5 minutes or so. I'll talk about using these microchambers that we create by surface modification of materials so that we can use these for developmental toxicity or screening cardiac defects with respect to drug exposure for early embryos. Okay, so Mark McCullough stole my thunder yesterday. He said this in the Q&A, which is this is Moore's Law, and some of those that are older in the audience would know there's the little Mac that's saying hello, right? You remember those commercials? The younger folks look up at the watch, but what you want to look at is this curve. Is what you're seeing is basically the increase in computational power increases by a factor of 2 about every 18 months, and that's what that curve shows you. And then, as was discussed yesterday, if you flip around more, you get Eroom's Law. And Eroom's Law has, I'll see if this, yep. Eroom's Law has, oops, okay, I'm going to avoid using I'm going to use a real pointer. Eroom's Law has this negative slope. If I can get that to go, negative slope. And what you see at that line is the cost of basically US dollars, $1 million per drug development. And you could see where that line's going. It's basically plummeting to zero, and the point was made that we won't be able to develop a drug with a reasonable cost without some change in how we basically do our analyses and drug screens. And we already see a couple of approaches of how that might occur, and I won't really belabor essentially the idea of where we can intervene in this process. But in our case, we don't think we're going to be upstream in the 10 to 15,000 compound range. We think there are adequate screens for that. Where we think we want to position organs on a chip technology is really in this mid-range, basically here, a couple of hundred drugs to screen, where we can give much better high content to the system in terms of analysis, as well as helping out with this process, which is clinical failure. So I like flames. Students, I use the flames to catch their attention. Maybe some of our colleagues that are a little older need flames to wake up. Okay, so when you see a flame, that's important. $5 billion average per drug development in US dollars. Duration is clear, 9 to 15 years. This is totally unacceptable. Part of the problem is, of course, we rely too heavily on animal models for this type of work. And even though animals want to behave like people, they are not people. It's already been discussed what their differences are, ion channels, differences in their essentially basal biochemistry and physiology. The big ticket, though, we see, even though heart and liver toxicity is a big issue with respect to where drugs fail, what is really interesting to us is this efficacy problem, which is if we can use these chips to predict efficacy, not just toxicity, because we think they are pretty good tox screens. Efficacy is where we can capture basically a movement forward. I think the question was asked, can we reduce the time to mark? And I would say my answer would have been yes. We can certainly reduce the time if we design better systems in which we can look at the problem. Okay, so human iPS cells has been introduced. You can bring in biological and racial and you might even say age diversity. We'll talk about whether personalized medicine is the way to go. So our paradigm is to build upon this concept, is to take the idea of basically human iPS cells, differentiate them into cardiomyocytes. And in our case, the cardiomyocytes, we'll talk about organization and structure in a moment, but this differentiation protocol is well established. We see the failure in the current way in which drug discovery is handled is this sort of artificial structure where the Parthenon or the house is missing a supporting pillar. And that's because we're too heavily reliant on these animal studies with their known problems. And so what we do is we add a second pillar, basically based on human iPS technology. And the advantage to that, and it's very clear we drew this as two distinct pillars because we don't want cross correlation or cross a comparison between the two systems, because if that was the case, if we were trying to prove that iPS cells could work as well as a predictive of drug success and efficacy, then why not just use the animal models to begin with? So we want an independent sort of FDA or regulatory body review of these kinds of systems. So flames, in our case, then what's the minimal organoid unit that we can use to make this kind of discovery? And this is nominally where we're moving. And so I think this community is well versed, but I just want to make sure we point out that, if we're just doing generic drug screening, then we can use wild type iPS cells and differentiate them to the cells of interest. But if we're interested in personalized medicine, then we need to know a couple of things. Basically, we need to know the patient's screen for acquired toxicity. This could be a patient that received positive or negative response to like cancer therapy, like docs. We need other aspects of confounding effects with these iPS cells. And we also, this is very important, we need to make these isogenic controls, and I'll hit this point again, because the isogenic control is the single genetic defect in that cell, which we can compare to now patient-derived iPS cells and determine the observations that we make. Are they related to the patient, or are they related to basically genetic defect only? So this is an important way to look at the problem. So sort of a little bit of extension of introduction is just a lot of folks working on this, including a beautiful talk that you just saw. What we see is that you can look at engineered heart tissue. Some of this work here is Chris Chen's work. You can start to think about how many of these can we get on a plate to make these scalable and to try to maybe approach kind of that Moore's law with respect to tissue engineering. And, we're thinking, well, maybe you can get 100 on a plate. That means they have to be small. But getting small, you can't sacrifice being predictive, right? And so we started our work a number of years ago with this idea, which is the heart has structure, and every student in material science would recognize this structure, what we call the process structure performance relationship, as a function of how they all affect the ultimate performance of a tissue, or in this case, I would say a material. Largely in biology, we've ignored that, and we haven't really thought about the structure when we make these sort of organoids or systems in vitro. And so the heart looks like this, and if you decellularized it, you would see kind of this structure where collagen fibers band in this kind of dimensional unit, about 150 microns. So this is a little bit larger than the width of a human hair. And this is basically the void where the cardiomyocyte was, and this is just collagen structure. They're not perfectly aligned, which is interesting. So this is a human tissue. But our hypothesis is that cell alignment and cell-cell contact in a 3D structure will promote essentially the kinds of things we're interested in. So here's where the microtechnology comes in. If this is a silicon wafer, just to give you scale, our cells sit in this little region here, just a very small strip within the chip. So it looks something like this. Like Melissa mentioned, we don't want a lot of pumps, so we just have one fluidic port for this particular chip, and then we collect it with a pipette tip, what's coming out of it, and I'll explain that in more detail. And so we have a central chamber that holds the cells. We microengineer this. You can see the dimensions. This is a mock blood vessel going down in these directions. These are little fenestrations that allow only for diffusion of the molecule into that central chamber. So it's a mock endothelial system, and I'll explain maybe in more detail why we think that's important. It only leads to diffusive transport, no actual flow, which is rather similar to how the cardiac cells would behave and be bathed. This is real time of fluorescein dye coming in, and you can just see within a couple of seconds the fluidic systems liberate dye into the center part. I'll describe what these little pillars are in a moment. You can even see the fenestrations as basically little railroad ties in the system. Okay, so what we want our system to do is to make a lot of measurements simultaneously real time. So we want real-time measurement of force, we want real-time measurement of electrophysiology, we want real-time measurement of things like contraction, which we can do, optically. And so we can also measure what's coming out of those media ports. And so the cells sit here, as I mentioned. We can do mass spec on whatever is flowing out of this. And then we can do destructive sampling as well. And then you'll see, of course, with the use of iPS cells, we can also use reporter lines. And that becomes pretty interesting for us. So this is what one system would look like. I'll go into this in a little more detail. Should be a movie playing, maybe it's not. But you get the idea. Cells are sitting in here. These are the electrodes. These are the pillars for measuring mechanical force. We used a differentiation protocol that was developed by Sean Palachek's lab at University of Wisconsin. I think some of you have seen this. This is the obligatory sheet meeting that you have to show if you do this kind of work. What I want to point out, though, by this slide is not really the methodology. We can go into that in detail later, in the gene expression that takes from pluripotent capacity to at least an immature cardiomyocyte. It's this cardiomyocyte population. About 75% of our cells are cardiomyocytes, about 25% are not. We've done different ways of looking at this based on Wolfram Zimmermann's work, showing that you need this non-myocyte population to get good tissue constructs. And so in our case, when we load our chip, we get something that looks like this, beating cells. The alignment forces this average motion. This is our x direction. Average motion in the x direction versus the y direction. You can look at the contraction relaxation velocities through an optical analysis software that we have written to give you this kind of information. And then you can, of course, do destructive sampling, which is just a confirmation that there's sarcomeric alignment in these kinds of systems. So I want to make this point very clear. So these little red arrows you're looking at are basically the vectors. They're spinning around like the hands on a watch because basically you're seeing movement of contraction and relaxation, and the optical software doesn't care whether it's relaxation or contraction. But I think if you look at this, it's very obvious. In a 2D, this is sort of, I'll use the term nilly-willy. I don't know if that translates up here, but it's not really coordinated. It's just sort of beating. It's a nice sheet beating. You can probably make some electrophys measurements. But it's very clear in this case, all the vectors are lining up going back and forth, because the cells are beating in this coordinated fashion. And so that's like the hallmark of what we're working on. We also use this GCAMP reporter, GCAMP6 cell line, so we can look at the calcium transient. And this is kind of cool. It's sort of like a blinking traffic light, right? So the cells are aligned. What you can't really see very well is with the speed is that the direction is kind of unidirectional in the calcium flux. And we can both compare the beating velocity and the fluorescence and actually map them right on top of one another at the go state, because they're both optical measurements. It's a little bit hard when you put in electrophysiology because you don't know where the start is. But in this case, electrophysiology with recording like electrodes. We can do force of contraction as well, and I won't really say much about this, except two points that are interesting to us, that we have good sensitivity. The actual forces that we measure and the stresses are similar to human tissue slices that have been obtained by various investigators. So we can get really the kinds of mechanics that one would observe with freshly harvested slices. So you want to see drug exposure. Here's the first example. If I didn't tell you which one had the drug, you should see that this one on the right is beating much faster, right? So he's given isopaternol. we calculate an IC, in this case, excuse me, an EC50 based on this, and this is just to show you how we're looking at this problem. This is the estimated therapeutic plasma concentration, so that's maybe where you might see drug dosing, and then this is the EC50, and that's just safety margin. And it's the ratio of these two that a drug company would use to decide whether it's a go or no-go moving that drug forward. This is obvious that basically this will have a nice safety factor, and if you look at the table of all the drugs we've tried, safety factor 180 is quite good. Typically, drug companies will look somewhere in the ratio of about 15 to 20 as a good safety factor. So that brings me to this example, verapamil. Verapamil is our favorite example with our chip, mainly because verapamil works on two different ion channels. It both prolongs and shortens the QT interval. You know how it's used for treatment better than I do. We get an IC50 curve that looks something like this. And if we do the sort of cartoon fly-in, we get a good safety margin. And the safety margin for us is about 18. And this is the real important part, probably the most important part of what I'll say this morning, which is if we took the same basically differentiation protocol, did cells 2D on a dish, we would get a safety factor of 3. In other words, it would be a no-go decision for this drug, verapamil, which is known clinically to have very few adverse effects. If we take embroid bodies, 30-day or 82-day, this might be an indication of maturity. You could see there's an improvement, but this is still in the danger zone, right? And so our 18 scales well with Escher Hagens in Germany, totally unrelated. We've never even met, but largely came out with the same number in the engineered heart tissue. And then pig, papillary muscle, and other systems come in with similar types of results. And I think that's good. And it's an example of maybe why you would spend the time to make a structured system. If we talk about personalized medicine, this has been introduced. You would use a patient with a certain disease state. In this case, this patient had long QT3 syndrome, had a QT interval about 500 clinically, sodium ion channel defect. We could monitor that on our chip now with the electrodes. We can see some aberrant effects, maybe EADs or DADs in the beat rate. If we gave that cell grouping, this system, verapamil, beat rate comes more in line, and we can plot that data as a function of basically arrhythmias related to the concentration of verapamil. And so this gets to the sort of patient-specific part where we could take this particular patient's cells and see how they would respond to not just verapamil, but other drugs that are known to try to treat long QT3 syndrome. Okay, I have a couple more minutes left, and I want to get to the next part. But you know, I leave this as a question mark. Organs on a chip, future of personalized medicine, patient-specific, disease-specific, reliable for drug testing, rapid, quantifiable. Like Melissa, we have a number of cells that we've all differentiated from the same IPS background, so we can start to make basically an autologous human on a chip. And we have a program funded to do fat and liver together. And so all the discussion about diabetes and heart, problems with essentially comorbidity or indications for those diseases are of interest to us. So our last part is thinking about how would we take this into the idea of using this for early heart development. And so for good reasons, women that are pregnant don't volunteer for clinical trials. These assays to test drugs are, to be polite, are just horrible. I mean, they really aren't very good predictors of how a drug might affect the developing fetus. I'm just showing you this video. I'm sort of running out of time, so I'll just do this quickly. This is a quail heart development outside or ex vivo from the shell, and now you're looking at the heart tube development and then early blebbing of basically the heart tube creating these trabeculations of early heart development and this rightward looping. So you can see it kind of just at the end. It sort of bulges out right about now. You'll see that. So, and then these are just lineage tracking of cells and how this happens. This is a very interesting process. I don't have time to go into in detail. It's not our work. It's work from colleagues. But we took that idea and said, okay, why don't we make a system in which we control regionally where cells are on the substrate surface. Chris Chen, myself, and many other groups have done this, including Peter Sansfer's group here. And in this case, the only difference is we're doing this on a current plastic platform that you use for cell culture. We use a polyethylene glycol thin film. We then use ionic charged gas, so that's not blood plasma, but basically oxygen plasma. It etches away through this mask, and this mask basically just leaves holes where proteins and then cells can be deposited. Very simple process. We developed this about 15 years ago. So all of these cells express pluripotency markers. What's interesting, if we then start the differentiation protocol with iPS cells in those circles, and their circles are ranging a couple hundred microns, so 400 to 800 microns, we see an outward migration of these cells to the perimeter. And so we get this annulus of high density, which also is correlated with some proliferation. And so you get differential states of these cells. And the short story is that the cells are at a higher density. They're under higher stress. They express markers of stress-related or mechanobiology events at the perimeter. Makes this beautiful little image where OCT4 is turned off late at the perimeter. Something interesting is going on here, which means the OCT4 is already turned off, and we're starting to see the differentiation. Row A actually scales basically radially in this fashion, meaning the cells at the perimeter are under some sort of higher stress or load, in a different biomechanical environment. And what that leads to are these cardiac microchambers. So they're actually hollow, and they're about 600 microns in diameter, and they have a height relationship. And we think these are good systems in which to start to study drug exposure to basically the developing heart. And so we were asked to do that. So we took thalidomide as the appropriate drug to look at. And essentially we can measure things that are geometric properties of the chamber. So its height, and then it's a half width at half maximum. These are just metrics to try to capture the geometric volume of what's been developed. And you can just see, no matter what we're really looking at in terms of the features, the control compared to the thalidomide exposure, you can see statistical significance in basically the alteration of the chamber. It turns out, if you really just look at this right here, you could see this is a nice sort of cross-section of the chamber, and this is really a dumpy little expression of cells that aren't really beating very well. They also have different contraction velocities, and they change the beat frequency as well when you have this drug exposure. So I think organs on a chip is not only good for adult personalized medicine, but possibly for embryonic personalized medicine, certainly for developmental toxicity screening, but also we think for studying early cardiac development that we may have a model largely based on material properties. And then I guess the advantage of going last and having your own computer is you can build upon what we really want to do. And I wish the first speaker in this session would review our grant. So this is an image from our grant showing basically bortezomib just to the wild type, and then with different defects in contractile proteins, and showing complete cessation of beat rate during this time of exposure and recapturing this. So we are very much interested in this idea of correlating our chip technology with cancer therapeutics as well. And then I'll just say there are a lot of growth opportunities. Yes, we're all here talking about medicine. I was giving a lecture at West Point with respect to organ storage. Army wants to figure out ways in which you can actually make better cryopreservation systems. My argument is if we can't do it on a chip, we certainly can't do it in a whole organ. And then there's other things that you can see, and we've discussed all these. And I'm just going over a minute or two. I need to thank, I just present the work. I have a very great team of postdocs. Anurag Mathur and Peter Larsk with the Heart on a Chip. Zen Ma, who's just starting his independent career at Syracuse University, he did all the work on the embryonic cardiac microchambers. A lot of this wouldn't be possible without Bruce Conklin and the collaboration that we have. This is San Francisco Bay, this is Berkeley, this is San Francisco. And then our whole liver program, which I didn't say anything about, is handled by Holger Willingbring. So thank you for your time, and I'd be happy to answer a question or two.